So quick review that viruses, and this is the simplest kind of virus, and it infects bacteria, called the bacteriophage. The basic stages are when they attach and inject their nucleic acids, they fall off. The nucleic acids are used as code to build more virus parts. Remember, the cell is doing all the work here. The virus just gets its code in there, and from there, it's the cell using the energy, the raw materials, doing all the work to build more virus parts until eventually, at some signal, those parts snap together, pop the cell open, and those things spew out ready to infect more cells. Any question about that basic cycle? So it turns out that, of course, as you get into viruses that infect more complicated living things, the viruses get new tricks. We'll talk about one of those tricks. We're going to skip another one, and I'll just kind of mention it. But you've probably heard the term retrovirus. HIV, the virus that causes AIDS, for example, is a retrovirus. And what retrovirus means is it's a virus that has RNA instead of DNA, which seems like not a very good strategy for a virus, because you know some stuff about RNA. Very versatile, very cool, but not very durable. It tends to get used and then broken down and recycled for parts. Theoretically, it shouldn't be the right thing to inject into a cell and have that cell make a whole bunch of virus parts. It wouldn't last that long. But the other thing you got to know is that all retroviruses come with and inject, along with their RNA, they inject a protein, an enzyme, called reverse transcriptase. For a sticker, what do you think reverse transcriptase does? So transcription is what? Transcription is taking DNA and making complementary RNA. Reverse transcriptase takes the viral RNA that just got its way into the cell and transcribes in reverse, making viral DNA from the viral RNA, and now the cell's in trouble, right? It's almost like a little Trojan horse, right? It's almost like a little sneak attack where, oh no, it's just RNA, no problem. Oh, now it's DNA. So it's another sneaky trick by viruses. So there's one trick. We'll just briefly mention the other one here in a minute. Meantime, something that's important to know about viruses, I think I've mentioned it, but they tend to be real specific. Viruses tend to not like to jump from one species to another. It's the exception rather than the rule. You know, you're not going to get your dog's flu virus. Your pet parrot's not going to get your cold. Turns out there are viruses, literally, that infect every single kind of living thing on Earth. Both kinds of bacteria, fungi, protists, plants, and animals all get viruses that are specific to them and tend to not jump. They also, in multicellular organisms, the viruses that affect those organisms tend to only like one or a couple of cell types. You know, the common cold belongs to the family of rhinoviruses. Do you know why they're called rhinoviruses? You know what rhino means? So dumb. Ella, are you pointing at your nose on purpose? Yeah. It means somebody thought that that thing was the rhinoceros. The rhinoceros. Yeah, that's what the root means. It means nose. And so it turns out that rhinoviruses only like to invade and replicate in the cells in your nose and throat and mucous membranes. That's why the cold is the way it is. It doesn't jump to other cells. And you've been evolving with the viruses that humans get. You know, humans have been evolving with those viruses for, you know, hundreds of thousands of years. But here's the problem, is that there's that every once in a while exception, right? Every once in a while, a virus will jump. Ebola, prime case, right? Jumped from, I don't know, monkeys, bats, something, and got into humans. Well, that's a problem because humans haven't been evolving with Ebola for very long. Right? We don't really have evolved defenses against it. Not only that, but because it jumped from a different species, that also makes it so that it doesn't really care much which kinds of your cells it invades. And when you got a virus that's a generalist like that, that is trouble. If it's invading like a lot of different human cell types, that's when you get situations like a hemorrhagic fever where you're, you know, everything's kind of falling apart. The pandemic strain of coronavirus, SARS-CoV-2. Is that it? 2? Number they put? Anyway, that's another example that we think it jumped from what? Bats. Yeah, we think it was a jumper from bats. And so the mammal, kind of pretty unrelated to humans as mammals go. And so, you know, that's why we call it a novel coronavirus. Novel means it's new to human immunity. And so we have real trouble with it, right? Maybe not 30% fatality rate type trouble, but, you know, the contagious nature of it meant it was a real problem. Killed a lot of people. The other virus trick that we usually learn about, I'm just going to mention, has to do with what we call the lytic and lysogenic cycle. Don't write this down. But the lytic cycle is the normal infective cycle that you already do. It's where the virus attaches, injects its RNA, and the thing pops eventually because there's thousands of virus parts. The lysogenic cycle is tricky because what it does is it injects, and then it just sits there. And it just kind of lays dormant in the, and it's just, remember, this is viral DNA that's in there. And so we say it remains temperate. And when the host cell divides, it also copies viral DNA. So what you wind up with is, and this is another thing that HIV does, it gets into your helper T cells, and then when those cells divide, they copy viral DNA as well. So it's pretty soon you've got this whole host of virally infected cells that aren't doing damage yet. And there's this signal that comes eventually where they all go lytic at once, they all do the thing you drew at once, and it's sort of a you know, almost like an ambush. This is a diagram that'll kind of bring that home, right? The DNA's in there now, but it's just waiting. And so as the cell divides, the viral DNA gets copied too, and some signal, and we're really not sure what the signal is, everything goes haywire. Great non-lethal example of that is, have you ever had a cold sore? You ever notice how fast those stupid things come on? Well, the answer to how that happens is that the DNA is already there in a cluster of your, you know, your lip cells or your inner cheek cells, and there's a signal that happens. Often it has to do with having some other virus, and suddenly you got a bunch of cells busting open, and you know, you notice it in first period, and by third period, like, huge sore. Anyway, that's another virus trick. But I really want to talk about, the last thing I want to talk about is prions. Prions are weird. Prions are one of those things that the first people to discover them, nobody believes them. Because, okay, you know, you got bacteria and protists and fungi as pathogens. That makes sense. They're living things that reproducing. You got viruses. Maybe they're kind of on the borderline of living things, but they have DNA. It makes sense what they do. Prions, they're just proteins. And you know proteins, those are just chains of amino acids folded up a certain way. That doesn't make sense. How could that be a pathogen? How could that actually cause a disease that could then be transmitted somehow? It took a long time to figure it out. And it's kind of an interesting story. So it turns out, all right, just humor me. Close your eyes. I want you to imagine all of your friends. Kind of start with your close circle of friends and then widen out maybe 1 degree to like the people you kind of see sometimes, hang out with sometimes. Now, out of all those people, can you pinpoint the one that if they're around, suddenly everybody's way more likely to get in trouble? Like if they're at a party, somebody's probably going to call the cops. You know that kid? Okay, open your eyes. And by the way, if you don't know that kid, it's you. So, you know, those kids that just influence people to do bad stuff, to kind of make poor decisions. Yeah, you know what I'm talking about? So that's prions. Prions turn out to look a lot like proteins that you make, but they're misfolded a little bit. They're folded wrong, so they won't do their job right. And not only that, somehow, and we're not totally sure how, they convince the proteins around them that are folded right to misfold too. They're like the peer pressure proteins. And the problem is that the proteins they look like have a pretty important job. They're the proteins that like hold your brain tissue together. So prion-caused diseases tend to be what we call degenerative nerve diseases, which is a nice way of saying they're diseases that make you go crazy and then die. So the story of how they figured it out, because the question was, okay, all right, researchers, we can buy that maybe if enough of these proteins build up in the brain, they convince the rest of the brain proteins to, you know, misfold and then the brain falls apart. All right, great. But like, how did they get there in the first place? How could brain proteins possibly be being transmitted somehow? And so to explain that, we just have to kind of go through what the diseases are. I'm not sure if this is the exact order they were discovered in, but it's close enough to tell the story. First of all, we have a disease called Kuru, which occurs only in Papua New Guinea, which is just north. It's an island. It's actually half of an island just north of Australia, off the coast of Australia. Only certain tribes in Papua New Guinea get this disease called Kuru, but it's a disease where in fairly rapid sequence, people go from kind of compulsive OCD type behavior to really crazy violent behavior to dying in not very long amount of time. Next, we have a disease called scrapie, which affects sheep in Scotland, or did. It's called scrapie because the compulsive behavior in the sheep in the early stages often involves them rubbing against like fence posts until they rub their skin off. If there's one you've heard of, it'd be this one, bovine spongiform encephalopathy, otherwise known as mad cow disease. Often abbreviated BSE. It's a mouthful, I know, but it tells you a lot about the disease. What does bovine mean? Having to do with cows, absolutely. Form means, shape, physical shape. Sponge means sponge. Encephalo, anybody know? That would be brain. Pathy just means disease. And so this is a cow disease that basically turns their brains spongy. And then it turned out that humans can get a version of mad cow disease, which we call Creutzfeldt-Jakob disease. CJC. And so by the time they've discovered all of these and made the connection that, oh, these are all caused by these weird juvenile delinquent brain proteins that make other proteins do bad things. What they can't find is what's the link? How do each of these populations get enough of the bad proteins, the peer pressure proteins in their brains to convince the other proteins to fall apart? And the way they find it is going all the way back to the beginning here with Kuru. And they go and they basically spend some time with the particular tribes where Kuru happens, and they discover one custom in particular that turns out to be the key to the puzzle. These guys, and it's not as crazy as it sounds on the surface. Basically, they believe that when an elder, particularly powerful or wise tribe member dies, first of all, they want to keep that wisdom and intelligence and power that person has, and they believe, not so crazily, that wisdom and power and intelligence is in that person's brain. And they don't want to lose that, and so they carefully remove the brain, and they section it up and divide it up among the tribe members, and they consume that brain tissue. And so there was the first answer of, okay, how are these things getting magnified and concentrated? How are the bad brain proteins making it into healthy people and getting to the point where there's enough of them to make the good proteins misfold? Well, there you go. That's how, in the case of Kuru. Well, okay, how do we connect this to these other 3 diseases? Well, in the case of these 2, it has to do with a not so healthy farming practice, but very widespread farming practice. You ever heard of a rendering plant? Basically, they take from the slaughterhouse and from animals that have just died and that they can't sell. They take the parts that still have tissue and stuff on them, and they throw them in a rendering plant and slowly heat them and melt them down until they kind of become sludge, and then they pipe that sludge over to where the grain is that they feed the animals, and they soak the grain in it. Why? Well, because we know that's the right stuff that the animals need, right? It's going to make them get big faster than anything else would. That's the logic. I'm not saying it's good, okay? But a farmer wants their animal to get to maturity to be able to be, killed for meat or whatever as fast as possible, and rendering the animal stuff into the feed is one way that can happen, right? Except that, of course, brains are part of that. And what you're doing every time as this disease takes hold is you're putting more and more prions into circulation, getting more and more prions into those brains. And so that was the answer to that. Now, how did humans get it? Let's just say you don't really want to know what's in your ground beef and your hot dogs, right? That's how and there haven't been a lot of human cases, but it was a scare at one point that this happened. And the thing about prions that makes them, there's several things that make them different, but one thing about them is that they're very resistant to heat. These proteins resist denaturing, so you can cook a burger and you still got the prions in there that'll do the damage. So the story, the link, the thing that all these cases had in common was, of course, a form of cannibalism. Weirdo. Good. Let me give you a guided reading for chapter 35. That's human immunity. That'll be the last real unit. This is the last real unit we'll do. Dissection kind of tacked on to the end.